let's get started. Hi, everybody. Thanks for coming. It is my great pleasure to introduce our speaker today for the Storer Lectureship, Dr. David Julius. Dr. Julius is the professor and chair of the Department of Physiology at UCSF, and he holds the Morris Herstein Chair in Molecular Biology and Medicine. He received his undergraduate degree at MIT and got his first research experience in the laboratory of Alexander Rich, studying how amino acids are attached to transfer RNA. And then he went to UC Berkeley for his graduate work, and he worked with Jeremy Thorner and Randy Scheckman, studying peptide hormone processing and secretion in yeast. And then he went to Columbia for his postdoctoral studies and joined the group of Richard Axel, where he developed novel expression cloning methods to identify genes encoding members of the serotonin receptor family. He joined UCSF in the faculty in 1989, and a major focus of his work has been to understand the molecular mechanisms of somatosensation and pain. His lab has taken advantage of the properties of unique natural products and environmental irritants and cloned a family of so-called TRP channels or TRP channels that you'll hear a lot about, which have completely transformed the field of somatosensation and pain. And this work has had immense practical implications, including in the treatment of pain, which we know is a major societal problem. Dr. Julius has won numerous prestigious awards for his research, including the Shaw Prize, the Prince of Asturias Prize, the Canada Gairdner International Award, and the Pisano Foundation Award, among many others. He is a member of the National Academy of Sciences, the National Academy of Medicine, and the American Academy of Arts and Sciences. Please join me in welcoming Dr. Julius.
Okay, thank you, John, and thanks for hosting me, and thanks to all of you for inviting me. As you can tell from the introduction, I've lived in the Bay Area now about half my life, and I've been to this campus many times, mostly as a graduate student, but I think this is the first time I've ever given a talk here, so it's really great to do that. And it's particularly nice to give a talk at a place where all you have to do is get in the car and drive there, so thanks for having me out today. So my lab is focused mostly on understanding sensory systems, and for those of you who I don't scare away today, tomorrow I'll be talking about a different type of sensory mechanism that we've been studying. But the main focus of our lab is to really understand this process of somatosensation, which we colloquially refer to as touch, and in particular the aspect of that relates to our ability to sense noxious or painful stimuli. So, and I put together this talk as sort of some details, but more of sort of a broad description of what we do, because I know it's a public lecture and it's supposed to be a broad audience. I know there may be some entomologists in here. I have some things about spiders in here as well, so hopefully there's something for everybody. All right. Okay, so why do we study pain mechanisms? Of course, pain really represents the main warning system that prevents you from experiencing tissue damage and injury and to initiate appropriate protective reflexes. And we all know that, but I think it's something that we take for granted. And in people in whom this system is not operating well, I'll put that here, namely people who have rare mutations that render the pain system inoperative or have diseases like diabetes or leprosy, those people are at greatly enhanced risk of injury, and you can easily identify, for example, a child whose pain system is not operative because they are missing digits, they may be running around on broken bones, they may get sand in their eyes in the playground and experience corneal abrasions because they just can't feel any of the discomfort or pain. So without this system, we are really at great risk. And I would say that among all our sensory systems, and I'm sorry if I offend any of you working on vision or olfaction or whatever, I think that this is the one that's most key to our well-being, to our health and survival. Okay? Now, the other interesting thing, of course, is that pain, also I think somewhat unusual for sensory systems, experiences or demonstrates great plasticity. And as we all know, especially those of us in the room over 50, this usually means in the enhanced direction. And that is such that wear and tear on your body and injuries can result in persistent pain syndromes whereby pain outlives its usefulness as an acute warning system and instead becomes chronic and debilitating. And a big goal in this field, of course, is to understand how this switch occurs at the cellular, molecular, and systems level with the aim of either preventing it or reversing it. And from a societal standpoint, of course, pain is the reason most of us go to the doctor. I don't know about you, but I hate going to see my physician for no reason, and the thing that always drives us in to see the doctor is we have pain. And in this country, you know, the numbers vary, but something like 1/3 of people, I think that's believable, experience some kind of persistent pain syndrome in their lifetime, and sometimes this just lasts for a few days or a few weeks, and sometimes it can last for months or years. And when people experience persistent pain, it can really upend their life, because then they start taking medications like opiates, and they basically, their whole sort of social interaction system and their ability to work really goes downhill. So we want to understand the basic rudiments of this. And the other thing I should point out is that, you know, when we talk about pain, it's sort of like talking about cancer. Pain we talk about as sort of a monolithic thing, a disease or a disorder, but it really probably represents a myriad and multiple different types of maladaptive changes to the nervous system, and manifests itself in different ways as musculoskeletal pain, arthritic pain, abdominal pain, headache pain, neuropathic pain, such as you experience when you have shingles or post-herpetic neuralgia. And so it's really a whole spectrum of these things. And I think what that means is that pharmacologically, they're also going to be, to some extent, distinct syndromes. And, you know, no one drug, even opiates, is effective at treating all different types, aspects of the pain experience. So we need to understand the molecular players, not just in pain per se, but in different models of pain and different mechanisms that drive different types of things that we describe as pain. Okay. So that's really the goal of what my lab and what this field is aiming towards. We focused mostly on the place where pain begins in this process called nociception, and that is the process whereby a subset of so-called primary afferent nerve fibers that innervate all receptive fields, cutaneous, your eyes, your mouth, visceral, and vascular target organs, how they detect environmental and endogenous changes that generate a noxious percept, this process of nociception. This is carried out mostly by a subset of nerve fiber types within primary afferent sensory ganglia, namely in dorsal root or trigeminal ganglion, that innervate everything from the neck down or the neck up, respectively. And that's the so-called small diameter unmyelinated C fibers and the medium diameter lightly myelinated A delta fibers. The larger cells are mostly devoted to the detection of things like innocuous touch or proprioception. So nociception and the ability to sense pain is mostly carried out by these two classes of afferents. And when they're activated, of course, they send a signal into the top part of the dorsal part of the spinal cord, where they synapse with ascending spinal thalamic tract neurons and convey this information up into the brain. So, and that's as much circuitry as I'm going to tell you about today. I know circuitry is hot, and as being sort of a neuroscientist now who doesn't work on circuitry, I'm sort of dating myself, but it is an important part of this, as it is any sensory system, to understand how the circuits are mediated. And I'll say a little bit about that, but mostly we're going to focus on transduction mechanisms at the primary afferent. All right, so now these sensory neurons, these so-called primary afferent nociceptors, the C and A delta neurons, they're actually quite fascinating and complex neurons. And as a group, they're tasked with a very complex job. And that is that they really, because they have to detect lots of stuff under lots of different circumstances. So first of all, they have to detect a wide array of acute, or what we call adequate stimuli, namely changes in environmental pressure and temperature, the presence of injurious chemical agents, environmental irritants, for example. And then when activated, these cells also, they're unusual in that they're called pseudo-unipolar cells, so they send information to the spinal cord, and then you perceive this as, you know, ouch, I did something to myself. But they also release transmitters antidromically. So they release transmitters from both ends of they sort of have two synapses in a way. And they release, in particular, peptides like CGRP and substance P. And then these act on cells in the periphery, in the area that's experiencing injury, in particular blood vessels, to elicit vasodilation and vascular leakage. And this draws into the area a whole bunch of circulating cells like neutrophils, mast cells, basophils, et cetera. And together with the blood vessels, then release or produce a panoply of other signaling molecules, such as histamine, serotonin, neurotrophins like nerve growth factor, proteases, bioactive lipids, ATP, and they reduce the local pH of the local tissue environment, so they produce a lot of protons. And collectively, all this stuff is referred to as the inflammatory soup because it's produced often during injuries associated with inflammation. And then what's amazing about these cells is that they have receptors for each and every component of this inflammatory soup on the surface. And when these inflammatory agents interact with the peripheral nerve terminal, they change the gain of the nerve terminal and enhance its sensitivity to things like temperature or touch. And this is why, when you injure yourself, you become hypersensitive to a subsequent stimulus. This is an essential part of the guarding and withdrawal effect response to tell you you've injured yourself, you need now to be hypersensitive to anything and protect that area from further injury and allow it to heal. So this is all part of the acute and sort of sensitization part of these nerve fibers. The problem is that when sensitized, this can get out of control and then lead to this cycle of what we call neurogenic inflammatory pain, where inflammation induces the release of these factors, and then sensitization of the nerve terminal, which causes further release of these factors, and so you can get stuck in this cycle. And one of the goals is to understand how this works so we can break that cycle. All right, so how do we study, so what are some of the questions in this field that I think have been important, over really the last, I don't know, many decades, but they remain interesting questions. How are noxious stimuli detected? How are all these things converted into the electrical signals of the nervous system, things like temperature, pressure, et cetera? How do these, what are the molecules and how do they transduce electrical signals into the primary afferent neuron? An important question psychophysically is how are sensitivity thresholds set? So why is it that if you touch something, let's say, below 43 degrees centigrade, that's fine, it may even feel nice and warm and comfortable, but if you go above that threshold, for most of us, except for a few mutants in the room, that will be perceived as noxiously hot. And the same thing's true for cold. You go below certain thresholds and it's not pleasant anymore, it's painful. So what are the molecular and biophysical mechanisms that determine those psychophysical thresholds? And then the most important question from a therapeutic point of view is how are those thresholds reset following injury? And I alluded to some of those mechanisms, but this is sort of a complex question. And then from a circuit point of view, how are different stimuli encoded? For all sensory percepts, we want to know how we can distinguish pressure from temperature, hot from cold, and what's the wiring diagram that allows us to do that. And then finally, can nociceptors themselves be targeted for analgesia? And this may sound like an obvious question, but there's been a fight in the field historically as to whether our perception of pain and our ability to discriminate things that are innocuous from noxious is really encoded at the periphery, or whether that's all encoded centrally, particularly in the dorsal horn of the spinal cord or higher. And there's been a lot of food fights over this, the so-called gate control theory versus the specificity theory. And I think data from our lab and many others now have suggested that there's coding at both levels, but there's certainly coding and discrimination at the level of the primary afferent nerve terminal, which says that yes, nociceptors are probably good targets for certain types of analgesic drugs. All right, so how do we study these things? So in my lab, years ago, we sort of turned to drugs to do this, namely using pharmacology, because for reasons I won't get into, historically, this is quite difficult area to identify main players molecularly. And I like this approach, which I sort of fell in love with when I was a student at Berkeley. Now remember, this is in the 70s, and so I really got interested in understanding how things like hallucinogens work, like psilocybin and all these natural products. How do they interact with the nervous system? And I thought this was fascinating because it's really, understanding how natural products work is really sort of the intersection of chemistry, anthropology, and in this case, neuroscience. And there's thousands of years that humans and other animals have had the ability to basically do all the field testing and come up with really interesting compounds that have important effects on us physiologically and behaviorally. So I call this pharmacology honed by evolution. And some of my favorite examples of this, for those of you who don't think about this, really relate to pain sensation, but also more generally to neuropharmacology. So one is the work of Solomon Snyder that influenced me a lot in the area of, say, serotonin receptors, but also in pain area, and that was his use of morphine and related analogs to discover opioid receptors, which are their endogenous target. Another example is the work of John Vane, who followed up on the well-known fact for millennia that willow bark produces an analgesic agent, which was identified as salicylate or related to aspirin. And he showed that the way this works is that aspirin suppresses the activity of cyclooxygenases, which of course produce pro-inflammatory prostaglandins. And then lastly, there's Raphael Mechoulam, who identified THC as the psychoactive ingredient in marijuana. And this was essential to, and he identified the so-called endocannabinoids, or the endogenous signaling molecules in our brain that are mimicked by THC. And this led to the identification of cannabinoid receptors that are the sites of action for both endogenous and exogenous cannabinoid drugs. So I think these are beautiful examples of sort of harnessing human development and anthropology and evolution of plants, coevolution of plants and animals, to really zero in on very useful pharmacological tools. And so we've sort of, and I should say that when you look at that previous slide, what's really impressive about it is that the two main arms of analgesic therapy that we still rely on today, namely opiates and nonsteroidal anti-inflammatories, come from this arc of discovery. Okay, so now in this case, this involves looking at natural products that suppress pain, and years ago we took a different tack and asked how natural products work that generate pain. And those also include many plant substances. The main one that we've looked at is capsaicin, the main pungent ingredient from chili peppers, which some people confuse with a taste response, because that's mostly how we take it in. But for those of you who ever chopped a chili pepper and forgotten that you did that and stuck your finger in your eye, that's not a taste response. Okay, so this is all pain, intense pain. And then there's also menthol, which we think of as a cooling ingredient, but it's at high concentrations is also an irritant. And then there are these other compounds, these reactive electrophiles, these isothiocyanates and thiosulfonates that you find in plants of the mustard and allium family. So when you're chopping an onion and you're like, having a lot of pain, that's because the allium in the onion it's volatile and it's gotten into the conjunctiva in your eye and activated sensory nerve fibers, CNA delta fibers, that then are triggered and you're in massive pain. So these compounds are known to generate pain. These are basically called mustard oils. And we wanted to understand how these work. And what's amazing about this is that although they've evolved quite independently in these plants, and presumably plants make them at least in part to defend themselves against mammalian predators like squirrels or deer, we've learned, we're humans, so we learn to like things that hurt us. But the way that they work, and this is shown for capsaicin, but it pertains to all the compounds, is of course they activate sensory nerve terminals, in this case in the trigeminal system. They send an action potential to the spinal cord, and you say, ouch, something happened. And then, as I said, they release transmitters from the peripheral terminals as well when activated. And that's why your lips become swollen and pouty when you eat a chili pepper, because that's neurogenic inflammation. Okay. And then what's more amazing about this is that although these have evolved quite independently, they've evolved not only to target the same subset of neurons, but they work through the same types of molecular transducers, namely members of this family of so-called TRP ion channels. These are non-selective ion channels that are permeable to cations, both mono and divalent cations, and when they let sodium in particular, it depolarizes the cell and sends an action potential to the spinal cord. Now, TRP channels weren't discovered in mammals first. They were discovered decades before we started working on them in the fly eye, through the work of Seymour Benzer and others, and Hardy and Minke and Zucker, et cetera. And in the fly eye, of course, light activates a rhodopsin, but in difference to our visual transduction pathway, which involves the cyclic nucleotide signaling pathway, this occurs through a G protein that couples to this enzyme called phospholipase C, which degrades PIP2 in the membrane and ends up activating this TRP channel. When sodium ions go through the TRP channel, the fly sees a flash of light. And of course, what PLC does is to chew up PIP2 and generate second messengers such as IP3 and release calcium from intracellular stores, as well as a variety of bioactive lipid messengers, including polyunsaturated fatty acids. And although we've known about this TRP channel for many, many years and this activity, what still remains at debate is how this channel is activated downstream of PIP2 cleavage. And this is relevant to what I'll tell you later, but genetics and physiology, mostly from Hardy and Minke, have suggested that the obligate step in activating this TRP channel is just to get rid of PIP2. So getting rid of PIP2 somehow disinhibits the channel. And then something else has to happen as a consequence of that, and it could be the production of a lipid second messenger or maybe intracellular acidification. But the whole mechanism that leads to the activation of TRP channels in fly eyes is still poorly understood. But the one thing I'll say is that this regulation of phosphoinositide lipid hydrolysis, as I'll show you, is key to understanding, I think, the physiology of these ion channels. So we now know that, that's just what I said, that in mammals, TRP channels actually represent a very large family. This wasn't discovered until really the genome was completed. And they represent the second largest family of ion channels, second only to potassium channels. There's great diversity in terms of their physiologic roles. And I would say that for most of these, we really still don't understand what roles they play. And that's in large part because we don't really have good pharmacological tools to manipulate them in vivo. There's little sequence homology. Whoa, what am I doing here? What happened? Oh, there's very little sequence homology within the families. They sort of expanded by this kind of radial evolutionary expansion. But as I said, many are modulated by PLC or PI signaling systems, as well as by calcium in some cases. And until recently, there's been a lack of high-resolution structural information to give us clues as to how these channels work. So let me tell you about what we've discovered about these channels in pain pathway, and then what we've discovered about how they work, including their interaction with lipids and what this may mean for drug design and development. All right, so we focused on 3 members of this family of about 30-odd TRP channels, and they're the ones that are activated by the natural products that I just talked to you about. TRPV1 is the receptor for capsaicin, TRPM8 is the receptor for menthol, and TRPA1 is the receptor for these sort of funky electrophilic irritants from, you know, wasabi and things like that. And today I'm just going to focus on these 2, mostly on this one, but on these 2. And the reason for that is that they're sort of related in the fact that they're very temperature-sensitive ion channels. So TRPV1, in addition to being activated by capsaicin, is activated by heat. And this just shows a slide of this channel expressed in a frog oocyte, where we've raised the temperature from, you know, from about room temperature up to into the noxious heat range, and then recorded current through this ion channel at the same time. And what you can see is that the channel's quiet until you broach this threshold of about 43 degrees, and then boom, it takes off. And the Q10 for this channel is really pretty amazing, much higher than the Q10 for most ion channels, not all, but most. So it has a very steep temperature coefficient of activation, which suggests that this is an important functional attribute of the channel. And the other thing is that this threshold of 43 degrees is interesting because, as I said, for most of us in this room, if I ask you when a heated surface or a pot of water would become noxiously hot, it would be after this threshold of about 43 to 45 degrees. Now, conversely, the menthol receptor, as you might imagine, cooling agents, is activated. Here we plotted it upside down when the temperature drops below about 25 or 26 degrees and the current begins to increase in this way. And so these are both temperature-activated ion channels, and their in vitro properties led us to suggest this model that they exist in the membrane of the primary afferent nerve terminal, where they serve as basically molecular thermometers. And when ambient temperature changes, they open respectively to hot or cold respectively, and elicit a sensation of experiencing something hot or cold. And when you come in contact with a natural product like capsaicin or menthol, what those do is to act as positive allosteric modulators of the channel, and they shift the channel opening to the midline, namely core body temperature, by lowering the heat threshold for TRPV1 and enhancing the temperature activation threshold for TRPM8, and therefore induce a psychophysical mimic of a hot or cold experience. Okay, so that's the model, and there's 2 things that that model suggests that are predicated on that. One is that these proteins are in some manner really molecular thermometers, that they either respond to temperature directly or at least in the right cellular environment. And 2, that we actually need these in the whole animal to experience changes in ambient temperature. So the first of these we've demonstrated, Erhu Kao in my lab, by expressing the channel heterologously, like in a hex cell or an insect cell, and purifying the protein to homogeneity, and then reconstituting it into a completely synthetic lipid bilayer, and then carrying out patch clamp electrophysiology on that reconstituted protein, where it's the only protein in the patch, and asking if it can respond to a heat stimulus, which we apply with an infrared laser. And when you do that, you can see that, in fact, you get these very beautiful outward currents that are characteristic to the channel in a very temperature-dependent way, where as you broach this threshold of about 43 degrees, boom, channel current takes off. And if you look at the biophysical properties of the channel, namely the temperature threshold of activation, its rate of activation, which is on the millisecond timescale, and then thermodynamic changes associated with that, what you see is that it very, very closely matches the channel that we've recorded from, either in whole cells or in sensory neurons for that matter. So I think this is nice evidence that, in fact, these channels are intrinsically temperature sensitive, and the only thing they need to sense temperature is the protein itself and perhaps its lipid milieu, okay? So that's about as reductionist as we can get in this system. Now, the other prediction from this is that if you don't have this receptor in your body, you have trouble discriminating ambient temperatures or temperature states around you in the environment. And the way we did this, of course, was several years ago. Diana Batista and Jan Siemens in my lab made a mouse that lacks tryptamate. So here's a wild-type mouse on a very simple behavioral assay where you put it on these two plates, a nice, warm, cozy plate and a cooler one. And without any training, like most of us, this animal knows that he wants to stay on the warm, cozy side, and he won't venture into this cooler side, no matter how long you keep him in this little apparatus. And he has free access to either side. But if you look at an animal in which the menthol receptor is knocked out, what you find is that this creature will wander across these plates and spend equal time on both surfaces. And we've done this over a range of temperature comparisons, and this animal really has trouble distinguishing a warm from a cold surface until you get down to really cold temperatures, like 0 degrees, where maybe tissue injury begins to play a role in that. So I think this shows at the behavioral level, this channel is required for our ability to sense ambient temperature. Now, for TRPV1, the story is a little bit more complicated because if you knock out TRPV1, you see pretty significant deficits in temperature sensation, but the animals are not completely heat insensitive, and there's been recent studies from other labs that suggest that there are 1 or 2 other TRP channels in the same neurons that contribute to temperature sensitivity. But TRPV1 plays a pretty dominant role in that. And you can do this experiment where you inject capsaicin into the intrathecal space, namely into the spinal cord, and what that does is it's neurotoxic at high concentration, so it ablates the central fibers of the sensory afferents, the C fibers that express TRPV1, so they go away. So if you activate this nerve fiber, you'll see inflammation in the periphery, neurogenic inflammation, but there's no behavioral response. It's the cord, not the cord, but the line has been cut to the spinal cord. And if you do this, what you find is that these animals are thermally analgesic. So if you take out all the cells that express TRPV1, they cannot sense heat. So if you put them on a hot plate, they go to what we call cutoff. You have to take them off the plate before they injure themselves. But if you ask if they can sense cold, that's normal. If they can sense pressure, that's normal. And the same thing's true of the of the TRPM8 knockout mice. They can't sense cold very well, but they have no trouble sensing heat. Okay, so this leads us to propose, in fact, that there is, or to say, we can say that there is, in fact, a lot of specificity at the periphery. There is coding at the level of the primary afferent nerve fiber. It's not just all centrally. And so therefore we can really sort of support this model of specificity that's been proposed, that there are labeled lines in the periphery, as is no surprise because this is how many other sensory systems work, okay? And this was proposed many years ago by Sherrington and then more recently by the late Ed Pearl, that cellular and behavioral specificities manifest at least to some degree at the primary afferent nerve terminal. In the case of hot or cold, this is manifest by having separate lines for hot or cold. And you can see this histologically because if you stain for TRPV1 and TRPM8 in the cell bodies in the dorsal root ganglia that give rise to these nerve fibers, they segregate into pretty much separate populations. Okay, so we think that, you know, identifying these receptors tells us something about how they work, but it also tells us something at least a little bit about coding in the periphery. And a main question in this field now is to ask what happens when all these sensory nerve fibers come into the spinal cord, and is this segregation of information maintained at the second synapse? Is it intermixed, interdigitated? How does that work? And how does coding, perceptual coding, get maintained as we go up through the central nervous system? And this is a question that we really don't, we meaning the field, have little understanding of. All right. Now, as I said, in addition to understanding mechanisms of acute pain, there's a real interest in understanding what happens after injury and how pain hypersensitivity is manifest. And that is, in particular, at least in the case of things we study, how does tissue injury and tissue inflammation lead to hypersensitivity to all things pressure and temperature? And in this regard, TRP channels, particularly TRPV1, has played an important role because in addition to being activated by temperature, it's a locus for modulation by many components of the inflammatory soup that I talked about. So there are many targets for those inflammatory agents on sensory nerve terminals, but TRPV1 is a target for a number of them. And this includes bioactive lipids like anandamide, one of the endogenous cannabinoids, lysophosphatidic acid or prostaglandins, extracellular protons, so local tissue acidosis is a big hallmark of tissue injury, and protons act on the channel, peptides like bradykinin and neurotrophins. And so TRPV1 is sort of a locus for integration for a lot of this information, and it's become a very interesting target to many people for treating inflammatory pain syndromes because it brings all this information together. All right? And that's really because it sort of exemplifies what we've come to love and know about TRP channels, that they're really polymodal signal integrators. And this has really been best perhaps exemplified by TRPV1, which, as I said, is activated not only by heat, but by various components of the inflammatory soup. And there are really two types of mechanisms by which inflammatory agents act on TRPV1. One class acts as direct allosteric modulators of the channel that bind directly to the channel and modulate its sensitivity to heat. And others, in analogy to the fly system, act on through second messenger systems by interacting with their own receptors, like bradykinin or extracellular nucleotides, or NGF on receptor tyrosine kinases, and tickling one or more signaling pathways, most notably phospholipase C, to modulate activity of this channel. And a big goal in the field is to understand what the biochemical downstream effects are that enhance the sensitivity of the channel to heat, particularly through these second messenger pathways, and also by allosteric modulation from these different agents. So that's a main goal because that's really central to understanding how one can drug these targets in an intelligent way. So this suggests that these channels, that this channel, and other TRP channels, but let's talk about this one for today, are good targets for the development of analgesic agents. So what's been the history of drugging these channels? And it turns out that now the pharmaceutical world has had some 20 years to develop compounds, and there are some very good compounds that interact with this channel, antagonists, and they do score pretty well as analgesics for a variety of inflammatory pain models like molar tooth extraction and bunionectomy and things like that. But they have sort of two what I call on-target side effects that have really been a little annoying to the pharmaceutical world. And one, as you might expect, is that patients have diminished ability to detect noxious heat. So here's someone on a placebo. Here are patients on increasing dose of this drug called ABT-102, which is a TRPV1 antagonist, and their ability to sense noxious heat is diminished because the stimulus has to be between 2 and 5 degrees higher for them to report that it's hot. Now that may not sound like a lot, but that's a lot when you're drinking a cup of hot coffee. So drug companies are worried that people will scald themselves on this, and I've heard that for some other antagonists, the diminishment in heat sensitivity is even greater. So, you know, that's something that you might expect from a drug that blocks a heat-activated channel. The other thing that was maybe a little less expected, but I think is also an on-target side effect, is that people experience a transient and usually minor, but sometimes more significant increase in their core body temperature. They become hypothermic. They report feeling a little feverish. And this may be because if you can't detect heat in your external world and you think it's cooler out than it is, your core systems then thermoregulate and tell you to increase thermogenesis, and your core body temperature goes up. Now, in most people, it's only about half a degree, and it lasts for a couple of days, but in some people, their temperature spikes 2 or 3 degrees, and that can be kind of dangerous. So what this suggests is that just blocking this channel by like putting a plug in it somewhere and turning it off is not the best strategy, and the best strategy is something more subtle, and that would be, for example, blocking the actions of different inflammatory mediators of the channel to block modulators but not turn the channel off entirely. All right. So the way to do that, I think, is to really understand what this channel looks like in 3 dimensions so we can be more intelligent about how we design drugs. So this has been a real problem in the field because nobody's had structures of TRP channels. They've been very tough to work with. But in the last 2 or 3 years, we and a lot of other people have made great progress in this. So let me tell you a little bit about what these channels look like. I haven't been keeping track of time. How are we okay? All right. So it's been presumed, just based on comparisons, that TRP channels look like they're very distant cousins, voltage-gated potassium channels, in that they have 6 transmembrane domains, where and 4 of these come together to form a homotetrameric channel. And the first 4 transmembrane domains in voltage-gated channels form this sort of extant region that serves as a voltage sensor domain that moves in the membrane when there's the electrochemical potentials changed. And the 5th and 6th transmembrane domains constitute or form the inside ion permeation pore. And for voltage-gated channels, as I said, when the membrane's depolarized, this region of the channel, particularly this so-called S4 domain, but this whole sort of extant region, moves in the electric field because it has highly charged residues that sense changes in membrane potential. This pulls on this thing called an S4, S5 linker that then opens the ion permeation gate between the 5th and 6th transmembrane domains, which would be seen at the very bottom of this hole down at the bottom of the channel. Here we are looking from outside cell to inside of the cell, and in cartoon version, that's how these channels work. Okay? And where trip channels are distinguished from KV channels, at least in part, is what hangs into the cytoplasm. There's a whole plethora of different things that hang into the cytoplasm, including protein-protein interaction domains, binding sites for regulatory molecules, et cetera. And this is really what sort of makes this channel family very diverse. So we've wanted to understand how do trip channels work, how do they compare to voltage-gated potassium channels, what can we learn from that, and can we learn anything in the case of TRPV1 about how all these allosteric modulators work to enhance its sensitivity to temperature. So for many years, we and others tried this by X-ray crystallography. It was kind of a nightmare, and we think that this is because these channels are very conformationally dynamic, and so it's hard to pack them into stable crystal lattices. And so we turned to this method with my friend and colleague Yifan Cheng at UCSF, where we used cryo-EM to do this. And I should just say that, you know, from structure function mutagenesis, we had some idea of where some things work. We knew that extracellular protons likely modulate residues in the pore domain, that capsaicin interacts somewhere in the channel around here, et cetera. So we had sort of very crude outline of where things work, and the question was, can we fit these into a three-dimensional model? All right, so Erhu Kao came to my lab, purified this protein, and here you can see beautiful monodispersed particles on an EM grid. Each one of these is, when I first looked at these, I thought, can this really be an ion channel with this little hole in the middle, a donut? But that's exactly what it turns out to be. And we use cryo-EM to solve the structure of these. And as many of you may know, or maybe a lot of you who don't do protein biochemistry, one of the beauties of cryo-EM that's really made this field explode in the last couple of years is that as opposed to X-ray crystallography, it's not a requirement that you have all the proteins locked into one particular conformation to form a crystal, and that they may in fact be dispersed on the grid in multiple conformational states. But thanks to the beautiful computational work of people like Scherz and others, one can computationally bin these different states, these different profiles into different states, and computationally assemble structures for conformationally distinct proteins. So this is a huge advantage. This is really what we as protein biochemists want. We want to be able to see a protein not just as a static picture, but as it goes through its life cycle of movements. So cryo-EM's turned out to be sort of a magic tool for this, among other things, and really solved our problem. So, the other thing is that this can really be enhanced if you have a protein like this one, where we really have some great advantages on pharmacologically. So if you have agonists, for example, like capsaicin, or there's another compound called resinifrotoxin. This is an extreme irritant from a flowering cactus that you find, for example, in the Mexican highlands. It's a member of the Euphorbia family. This is the most irritating substance known to mankind, but like capsaicin, it has this vanyl ring, so it's also a vanilloid compound. It's basically a super capsaicin agonist. If you get this stuff in your eye, it's not fun. So we always, when we get a new bottle of this, when we have a new student in the lab, and we get some of this in, we'll give them the can just to see them, and say, could you aliquot this out? And then they start taking, and it's like a box with a can inside another can and another can, and then they sort of freak out. Then we do it for them, but it's kind of a hazing experience. And then, so plants use these to defend themselves, but plants are not the only thing on the planet that has to defend themselves from predators. There's also spiders and scorpions and things like that, and they use their own form of chemical warfare to do this. And we've discovered, Chris Bolin, who was a student in my lab, discovered a toxin from this spider called a earth tiger tarantula, or a Chinese bird spider that's found in southern China, and it makes a peptide toxin that's also a super agonist for this channel. So when it bites you and you're in pain, that's part of the reason why. And so we've been able to use all of these tools, agonists, all agonists, to push this equilibrium to the open state and to really be able to use cryo-EM to look at these structures. So what we've learned from this is that the channel does indeed look like a voltage-gated channel overall. It's a tetramer that forms this sort of pinwheel-like structure where you have these extant voltage sensor-like domains and an internal ion permeation core. I won't go into all the details of this channel because I don't have time, but we figured out, for example, what these anchoring repeats do in the context of TRPV1, that they help hold the subunits together, and other sort of major structural characteristics. What I want to focus on really more is what happens inside the channel, namely in the pore, and what we've learned about that and how it's related to what we think are the special properties of trip channels. So unlike voltage-gated channels, what we found is that there are two constriction sites, if you look down the central axis of the channel, that apparently need to be open for ions to flow through, and this suggests that there's a very complex sort of dual gating mechanism in these channels that must be, one, it's a selectivity filter where ion selectivity is determined, but aside from being a filter, it's also a gate, and one in a lower gate that corresponds to the sort of gate that you would see in a voltage-gated potassium channel. And this suggested to us that, that in fact, this might underlie the very complex regulation that we see of trip channels, particularly TRPV1, by inflammatory mediators that likely act both up here and down here to regulate the channel, and that this confers these channels with a lot of pharmacological flexibility for being able to appreciate a wide range of physiological stimuli. But the only way to know that is to visualize the channel not only in its closed state, but in its open state. So we did this trick of giving the channel its favorite agonist, namely resiniferatoxin, and then throwing the spider toxin on top to lock it into an open configuration. And if these are really operable gates, they should be opened. So we're able to do that, and here's the structure of TRPV1 with this resiniferatoxin, this capsaicin analog, locked into its vanilloid binding site, and then you can see this spider toxin docked on top of the channel. And what the spider toxin does is, it's not an agonist per se, but it's a gating modifier, so it waits for resiniferatoxin to open the channel, and then it goes in and it traps it in the open state. And lo and behold, when we did that, we can see that these restrictions are fully open, indicating that these are not just selectivity filter and lower gate, but they're both operable gates. And again, this suggests a mechanism whereby this channel is able to appreciate this rich physiologic modulation, because you have two regions of modulation by things like extracellular protons and spider toxins, as well as down here, not only by plant-derived agents like capsaicin, but also bioactive lipids that we believe mimic the actions of vanilloid compounds. And in the next few slides, I'll show you how we think that works. All right, so let me just say briefly that comparison to potassium channels reveals a lot of similarities, but it reveals some important differences as well. And that is that in voltage-gated channels, as I told you, there's a lot of movements around the so-called S1 to S4 domain. This is where voltage sensing occurs. In TRPV1, what we found is that that's a pretty static part of the machinery. It doesn't really move very much. And instead, where we see major conformational changes are down here by the lower gate, as you might expect, and then up here in the upper gate region, in the so-called extracellular domain. And this is, I think, explains why, evolutionarily, spider toxins have targeted these different regions of the channel. So Rod McKinnon and Kenton Schwartz identified years ago a number of spider toxins that block the activation of voltage-gated potassium channels, and they work by sitting on top of this voltage sensor domain, as you might imagine, because that's the functionally most salient part of the channel. That's where many of the conformational changes are occurring in a stimulus-dependent manner. Instead, what we find is that the toxin for TRPV1 binds up here. So I think this is why evolutionarily these different toxins have evolved to attack these different parts of the channel. And I think this is sort of chemical confirmation that the conformational changes we see are really occurring in this region, and they're physiologically relevant. All right, so we can look at movements in the pore. I won't go through this in detail because I'm running late. And we can understand how the toxin works to trap the channel in its open conformation by visualizing ionic interactions and molecular interactions between the toxin and the channel. And this tells us a lot about this site. The other thing it tells us is how things like extracellular protons work. And this is exciting to us because it's really the first glimpse of understanding how a natural pro-analgesic agent that we generate in the course of injury works to activate or enhance activity of this channel. And what Sven Hjort in my lab showed a number of years ago is that there's a glutamate residue in this upper gate region that undergoes a lot of conformational changes in our structure. That's the main locus for titration by extracellular protons. And if you look at the structures in the closed and the open state, in the closed state, what you see is that this glutamate residue is held close to these by these main chain interactions with this aspartate and tyrosine residue in a neighboring helix. And in the open state, these residues have been pulled quite far apart, meaning that these bonds have to be broken. And the way that we think extracellular protons enhance heat sensitivity to the channel is basically to salt out these interactions and pre-break ionic interactions that have to occur in the action of channel gating. And so they're basically enhancing channel gating by heat because they're pre-breaking these bonds that have to be broken during the gating process. And that's why low pH not only shifts the threshold of heat sensitivity back towards body temperature, but also enhances channel currents at superthreshold temperatures. And you can see this decrease in heat dependence, and that's probably because they're pre-breaking bonds that normally the heating process would have to break to open this channel. All right, so that tells us something about a really good drug site that can be manipulated to prevent sensitization of the channel by, say, acid. Now, the last thing that I want to say is that we've had a lot of fun looking at the structures of these channels in detergents and in other synthetic media, but these channels live in lipid bilayers, and what we really want to do is understand how they look and how they behave in lipid bilayers. And cryo-EM turns out to be a great method to do this if you combine it with this wonderful preparation developed by Sliger and colleagues at the University of Illinois called lipid nanodiscs. These are like little hockey puck-like structures that you can form in vitro by taking lipids and mixing them together with membrane scaffold proteins, which are basically derivatives of apolipoproteins, and they spontaneously form these little kind of these little hockey pucks, and you can insert membrane proteins of a certain size into them. And since they're single particles, you can use cryo-EM to look at their structure. And Yuan Gao, a student with Yifan and myself, did this and was able to insert TRPV1 into these lipid nanodiscs. And here you can see TRPV1 floating inside one of these lipid nanodiscs. And in fact, this really stabilized the protein even more, and he was able to get a very beautiful structure below 3 angstrom resolution in this nanodisc. And here it is. This little fuzzy stuff on the outside is a density of the nanodisc. That's the resiniferous toxin, again, packed into its pocket. Here's the spider toxin, and you can already tell that the densities are much more highly resolved. And the great thing about it is that you can also see the density for the phospholipid head groups in the membrane, so you no longer just have to draw dotted lines. We think the membrane is here. We know exactly where the membrane is. And the most important thing from this is that it allowed us to see really intimate interactions between a channel and membrane lipids. And for channels like this that are regulated, where regulation by membrane lipids is so important to their physiologic activity, this is really a great attribute. So I'll just show you one last thing about this, and that is that if you look at the resiniferous toxin binding site here, first of all, we could resolve this molecule with very good density. This is one of the first instances in which cryo-EM was able to really visualize the interaction of a small molecule with a protein. So for people interested in drug design, I think it finally really sold them on the idea that you don't have to necessarily use X-ray crystallography for that. But the other thing that we found out is that this site, when not occupied by resiniferous toxin in the apo site, is occupied by another density. And what we could do is to find out that that density actually corresponds, to a phosphoinositide lipid. So when there's no vanilloid agonist in the pocket, that pocket's occupied by a PI or PIP2 type molecule. You can see that there are nice charge residues in the area that can stabilize it. And this gave us some first structural insights into how PIP2 and other phosphoinositide lipids could modulate the activity of these channels. And the model that we've come up with for activation is that in the apo state, PIP2 is nestled into this pocket, and it's stabilized by interactions with this arginine residue and a glutamate residue. When resiniferous toxin comes in, or capsaicin, it kicks that out. And although it occupies the pocket in a somewhat higher latitude, and it frees up space and allows these residues to come together, this arginine and glutamate residue, to form ionic interactions. And when it does that, it pulls this S4-S5 linker that then pulls on the channel pore and allows it to open and allows ions to go through. So basically, this is a mechanism of displacement of an endogenous modulator of the channel that then allows the channel to open. And if you use an inverse agonist that can occupy this spot but doesn't activate the channel, it displaces PIP2, but it doesn't allow these residues to come together. So this begins to give us an understanding not only of how hot chili peppers work, but how phosphoinositide lipids might modulate this channel and possibly other TRP channels. And again, it suggests and identifies an important allosteric site that one could use to modulate activity of the channel without necessarily shutting it down completely. So in conclusion, I think natural products have really provided a great window into molecular mechanisms of somatic sensation and pain by allowing us to identify some key players in this area. As in all sensory systems, these channels and transduction elements, receptors, really provide molecular genetic markers for marking functionally distinct cellular populations and beginning to understand how coding works and how perception is achieved through coding specificity. And the TRPV1 structures really reveal architectural features that underlie the physiologic modulation by a number of endogenous agents. And this sort of dual gating mechanism probably helps us, well, does help us understand how these channels act as polymodal signal integrators to modulate neuronal sensitivity in sort of a complex changing physiologic environment. And at the level of TRP channels, there's been a lot of fights over the years as to what PIP2 does to these channels and how they work. My guess is that the interaction of PIP2 with TRP channels is going to be different for many different TRP channels, but in the case of TRPV1, I think it resolves some questions as to whether PIP2 acts as a negative or positive allosteric modulator. It really acts as both. It has to be in the channel for the channel to be in a ground state to be activated, but then you got to kick it out for the channel to be maximally activated. So these sorts of studies are beginning to tell us about some of the details and complexities about how these channels are regulated by physiological processes that regulate membrane lipids. And then finally, I think all these channels will actually, in the end, we hope, be targets for a number of analgesics, particularly those that treat a variety of inflammatory pain syndromes of a wide variety. All right, and then finally, I think this lists almost everybody who's ever been in my lab, but I just want to point out a few people who were key: Mike Caterina, Dave McKemie, and Werner Neuhauser for originally identifying TRPV1 and TRPM8 and its relationship to temperature sensitivity. Wai-Hu Chuang and Julio Cordero, Diana Batista, and Makoto Tominaga in particular for understanding the relationship of this channel to modulation by inflammatory mediators. And then Chris Bolen in particular, together with Avi Priel and Jan Siemens, for developing many spider toxins and other things that most of which I haven't had time to talk about, that have turned out to be really key pharmacological agents for manipulating these channels in vivo and in vitro, and particularly for structural studies. And then finally, Erhu Kao, Candace Paulson, and Yuan Gao for structural work on TRP channels using cryo-EM. And I've had a great pleasure of having some long-term collaborators at UCSF, Alan Bassbaum as my guru in anatomy and pain behavior, and Yifan Cheng, my partner in structural biology, together with Mao-Fu Liao, now at Harvard, who helped us with the TRPV1 work. And then finally, thanks to all of you for paying your taxes, because all the work in my lab is supported by the NIH. All right, thank you very much. Okay, I droned on for a while, but I'm happy to take questions if anybody has it. Yeah.
I was wondering what modulates the level of pain. So if you have more capsaicin, it's hard. Are you activating more receptors?
I think you're just activating more receptors and more nerve fibers. Yeah. It's a Scoville units. You know this Scoville pain? It's directly proportional to the concentration of capsaicin. Yeah. Yeah.
So you said that the S1 to S4 region compared to the conjugated plasma channel is more static. So from the structure comparison, can you tell the reason why this part in the channel is more static?
Yeah, I skipped over that. I had it in there, but Well, first of all, there are no positive charges in that region that would, in terms of voltage sensitivity. So there's some voltage sensitivity to trip channels, you may know, and everyone's always wondered how that's manifest. We really still don't understand that, but it's not through this region because there's no charges in there, it doesn't move. And if you look, in voltage-gated channels, those charges, the positive charges, arginines, interact with negative charges to, so this is sort of charge transfer motif that allows movement to occur in an energetically favorable manner. And in the trip channel, what we find instead is that region is just packed with hydrophobic residues. So it's totally different. It's the similar sort of structure, but it's held together in a very different way. Now, it could be that, for example, with heat, which likes to operate on hydrophobic interactions, that does move. And again, did get a chance in this lecture, but there are many more structures of trip channels now. And I think, and they all sort of move in somewhat different ways. But as far as I know, there hasn't been much movement seen in this region of trip channels. So I think it's going to turn out to be somewhat more static and play less of a role in gating than the movements that we see in the outer pore region and in the lower gate. And I should also say that trip V1, we got sort of lucky coming out of the gate because it's still one of the only trip channels, and really one of the only channels, where you can see the channel in multiple functionally relevant conformational states. And that's beginning to be true for more channels. For like trip M2, there's a little bit of that. For many trip channels now, there's like one static structure. You don't know whether it's closed, partially open, desensitized. And so it's really hard to make conclusions about what's moving conformationally because you don't see anything moving anywhere, really. So I think we have to wait until we can get more structures and more reagents that allow us to see these channels in both open and closed states. Yeah.
So with your phosphoinositide in the structure at the end,
Yeah.
you're saying you think it has to get out of the way for the channel to open?
Well, yeah, the reason being that when we look at the vanilloid compounds, they displace the PIP2. There's either PIP2 or there's capsaicin or resinoftoxin. They're mutually exclusive. And when you look at the changes that occur in terms of the side chain interactions, the side chain interactions that we think that occur that allow this S4, S5 linker to pull on the gate to open it up cannot occur when PIP2 or PIP, we don't know if it's PIP2, it could just be phosphoinositide, just PI, does not allow those residues to come together. So I think that the PIP, or the phosphoinositide lipid, is sitting there as a wedge and preventing the movements from occurring that are needed to occur to allow the lower gate to open.
And yeah, that makes sense. And with that, you also said it needs to be there. It's also
Yeah.
a positive.
Right, so mostly I say that because if there are other people here who've worked on trip channels, they'll start throwing stuff at me because there are a lot of people who have said, particularly for trip V1, that phosphoinositide lipids are required for the channel to open. And we've always said, based on mutagenesis studies, that it's a negative regulator of the channel, and that when you get rid of it, you disinhibit the channel and enhance its opening. So those are two sort of diametrically opposed models. But I think this structure makes those things whole. And I think the reason that phosphoinositide lipids are required for the channel to open is because I think they have to be in the channel in the closed state, maybe to stabilize it, that there has to be something in that pocket. We've never seen it empty. And we're trying to see if it can be empty, like by seeing if you have a spider toxin on top holding it, is that empty? But we've never seen it empty. There's always either a lipid in there or another agonist like capsaicin. And so, what we imagine is that the lipid has to be there for the channel to be in its fully closed, stable state, ready to open, and that then to be open, the lipid has to be ejected by something, okay? And how heat works in that context, we don't know. But that would sort of make whole of people's electrophysiological observations over the years. But that's just maybe our way of having a parsimonious explanation. But it's somewhat consistent, at least with the limited number of structures that we've seen. Yeah.
Is the reason a spatial cue?
Sorry?
Why PI lipids? Why PEP lipids?
Oh, because they can be regulated downstream of PLC signaling pathways. So it's a, huh? Oh, are in the pocket?
Yeah.
Well, we can't tell because we haven't had the resolution. We can't see everything on the head on the inositol ring, so we don't know if it's mono, di, or triphosphorylated. So we're trying to figure that out now. But in reconstitution experiments in synthetic lipid bilayers, we know that if we supply different types of phosphoinositide lipids to the membrane, we can shift channel activity by a number of phosphoinositide lipids ranging from PI to PI4,5 bisphosphate. So, but presumably those lipids occupy that pocket because ultimately they're substrates downstream of PLC signaling pathways, which again would explain, you know, how bradykinin and other things that activate PLC pathways enhance sensitivity of the channel to heat, which is another reason that I never really could buy the idea that PIP2 is required for channel activation without having an effect, any other effect, because how else could you explain that activating a PLC-coupled receptor enhances channel function if it's chewing up PIP2? So I think this model sort of tries to make sense of those physiological constraints.
You mentioned pressure. Does pressure also work on the system?
Not very much. There are other channels that people like Artem Potapudi and others have looked at, these piezo channels and others that likely are the main mediators for pressure sensation, at least for light touch. What mediates sensitivity to noxious pressure that causes pain is still somewhat mysterious.
So could the requirement for PIP2 means that when you take it away, which is usually a major blow to the cell, whenever it takes a little while, could that then cause maybe brief activation of PIP channels and then a desensitization? Have you seen a desensitized?
Yeah, so you can actually turn over the PIP2 in the, I mean, if you look at like cardiac myocytes and you activate a PLC-coupled receptor, it can take out a lot of PIP2 very quickly. Will it enhance channel activation? Well, you know, if you're, say, recording at room temperature and you activate a PLC-coupled receptor, you'll see TRPV1 currents, you know, begin to emerge at room temperature or a little bit above that. So what it does is it enhances sensitivity of the channel to capsaicin, to heat, to extracellular protons. So it basically shifts the threshold of activation by any stimulus left. So you'll begin to see, you know, what you would categorize as spontaneous channel activity after you activate a PLC-coupled receptor. And then, yes, the channel will eventually desensitize in a calcium-dependent manner. And the mechanism for calcium-dependent desensitization is not well understood. It may involve calmodulin, but that's not clear. Yeah. It does involve calmodulin for some other TRP channels that are calcium.